All right, welcome everyone. I'm taking over the intro duties from Jill Blumenthal, who's on her parental leave. So everything's going well there. So I'm pleased to introduce Steve Shoptaw. He's a clinical psychologist and professor in the departments of family medicine and psychiatry and biobehavioral sciences at UCLA. He directs three research centers at UCLA currently and receives support from NIH and other funders. Dr. Shoptaw's works in the US and globally lead research, clinical, and policy efforts that bring novel and high impact strategies to persons affected by addictions and infectious diseases. So, Steve, welcome.
Thanks,
Um,
Tim.
so
Appreciate you
it's
asking
about
me.
45 to 50 minutes. So there's, I'm sure, going to be robust Q&A. So please take it away. I'll monitor the chat for you.
Thanks, Tim. And by the way, I like chatting. So, like, if there's questions that come up, stop me, okay? Because and I have a short presentation today just for the purpose of talking. So,
Perfect.
thanks to you and to Jill and Marvin for organizing and asking me up for the high rounds today. I have a new talk. This is very fun for me. I do have disclosures. I get research, clinical supplies because I do medications research, and we also we'll be talking about Clear Scientific, where I provide consultant services. So before I start talking, this is an unprecedented time. Check the New York Times today. People are abandoned in the middle of trials with medications on board and devices on board. This is craziness. 178,000 people with HIV in India. 8,000 are kids with no support despite the waiver. So these are crazy times. We've never seen these kinds of times. The advantage of crisis, though, is that we have opportunities to respond and to and to remake our systems. And I'm hoping that we'll take some time at the end as we talk about the systems that we have and the way they can be improved. I have just a few items that I think is important as we move through this period, and especially starting Friday this afternoon and into Monday when NIH comes off pause, is to call your representative and senators to do self-care. Make sure you take time. This is a marathon, not a sprint. Be kind, always be kind, pay attention, and recognize that careers will be and already are being damaged. Finally, people will die, and HIV and STD incidents will increase. So, with that happy thought, I do want to point out that we have a very siloed approach to care. And so this is a really good time now to start building the architect or the plans, getting together with the architects of how we might want to see the system remade. Because there's not enough medication in the world to treat ourselves out of the HIV epidemic, out of the addiction problem, and with the problems with mental health and trauma. So, moving toward whole person strategies and treatments that involve different parts of expertise that people have in the same person is really helpful. And it may help us build and move beyond this inefficient system that we have, where we have to have catwalks from mental health to addiction and from addiction to HIV and STIs. This is an opportunity time, and I think it's helpful to just remember. In a different time, before this current setting we're in, I had a good idea about where we need to focus in terms of controlling HIV and addressing addiction and comorbid conditions in this country. I'm not as sure now, but this is still a good marker, a good model, which is that for about 80% of the population, standard biomedical, behavioral, and risk reduction approaches to HIV care and prevention are effective. That remaining 15 to 20% is where we need to focus. That is what I would say before. That is the next decade of work in HIV prevention and care, is that 15 to 20%, because they will always and perpetually provide viremia needed in the population level for forward transmission. So what we know about this, though, is that there is the erosion or unavailability of health resources. That is even more threatened today on a global level and maybe even in our US. We have the effect of substance use and mental health disorders that are ongoing in the people who are poor and have many disparities. And then we also have poverty operating, and that is the fuel for outbreaks of disease. Um, you know, I I think that this is an important model to keep in mind because as we deal with the problems that are going to be facing us in the next few days, and weeks and months, that we need to remember that our folks who are at highest risk are in these groups. They're poor, they have substance use, mental health disorders, and they don't have access easily to health resources. So in the interest of the banned word of equity, I would remind us that we this is where our patients live who are most in need and where disease actually is found. So lately, because I'm old, I start remembering why it is I got involved in this business in the first place. And I remember that in 1992, the purple part here, the overall mortality rate of men who have sex with men in Los Angeles, largely these are the men who are dying. The HIV had a 72% mortality rate. That's an important reminder coming out of the COVID era, where there's less than a 1% mortality rate, that this is where we started. In 1992, when I entered the field as a psychologist, I was like, this is a place where I should work. This is a serious problem. I have some ideas about what it might mean to get involved, but I'm a psychologist, not a physician. And at the time, there was really nothing that psychologists could do to render any sort of relief. And so I met with friends. On the top here is Kathy Reback, one of my work spouses, but over the past 30 years has been really important, who met with me at the 101 coffee shop in Hollywood. And we talked about the ways in which methamphetamine and HIV are linked. And she was one of the first to see that among gay men who were using methamphetamine, HIV was almost 1 to 1, which led to her preparing this ethnography. Still a good read. It's still consistent and current in Los Angeles. All of this work was funded initially by Ferd Egan, who was a man living with HIV who died. He was the LA City AIDS coordinator for a while. But it's important at these times in particular to remind ourselves why we went into this field to begin with, and to remember what it is we know. We know that methamphetamine in infectious disease settings and in clinical settings, we know that methamphetamine has functions that are important to the people who use the drug. Particularly with men who have sex with men, it allows a type of sex that's not available to them when they're not using the drug. For shift workers, as we go clockwise around the pictures here, methamphetamine helps you be able to work 2 jobs and still be functional. For people who are motorcycle riders, gang members, methamphetamine organizes behaviors and social interactions within these groups. Among women, methamphetamine helps them remain thin and being able to do all their household responsibilities and still want to have sex with their husbands or their spouses. Among the rural youth and rural and youth folks, methamphetamine helps time pass. And so they use, folks out there will use methamphetamine for that purpose. And among the homeless, this is a new graphic that I use, and I describe the functions of methamphetamine in people's lives. Among people on the street, at night is the most vulnerable time. So folks on the street will use methamphetamine as a way to stay awake at night and be able to sleep during the day. You can see here the physical and psychological effects. I'm not going to spend time with those, but all the greens are good and all the reds are good too. So it's a drug that fits a lot of people's needs. And I think it's always important to remember that when you take that drug off board, using a treatment or jail or whatever, we have the person experiences a hole in their behavior repertoire that we need to address as clinicians and as researchers. In terms of what's happening with prevalence, methamphetamine tends to be fairly consistently prevalent in the population level. You can see here that the blue is cocaine, the yellow is stimulant misuse, and the red is methamphetamine use. And you can see that it's about 9.7 million people who are adults who in the past year misused stimulants. So a lot of folks, but not a whole lot of folks. And there's a fairly good allegiance to the type of stimulants that people use. There is a breakdown in terms of showing that Asian folks have lower prevalence than others in the population. And that if we look at the National Survey on Drug Use and Health, methamphetamine prevalence, like I say, that's the leftmost 5 bars, is fairly fairly stable. But if you look at meth use disorder, you can see this nice increase here that's been going on between 2015 and 2019. This is where is the latest data showing. And it really shows that the higher meth use and meth use disorder co-occurs with people who are using other drugs, particularly LSD, hallucinogens, stimulants, and cannabis. So the question is, what to do and what's happening. We know that there's some new things going on, and I'm not going to spend a lot of time with this, but you can see here that the fentanyl bar is blue, and you can see how in 2015 really it takes off. And coke takes off with fentanyl, where at current rates we have about as many people dying from fentanyl without stimulants as with stimulants, which is a new thing. So if you look at this graphic over here, you may be wondering what this is about. And for folks who are treating patients who are using methamphetamine, methamphetamine is produced often in the same places or consistent located nearby, or is managed by folks who also either make or sell fentanyl. So we've all heard stories of people who use methamphetamine finding their way to being uh, fentanyl affected. So one of the things that we know about here is there's this hotline here. It's called Never Use Alone. It's neverusealone.com, and this is the number. You can dial this number 24/7. If you're a person using methamphetamine or fentanyl, they will sit with you while you're using and make sure you don't overdose. So that's one thing to think about in terms of advancing practice today from the talk, is if you have folks who are actively using, or you have a suspicion that they're using methamphetamine, just to give them this resource so that as they're using, if there is fentanyl involved or some other drug that's adulterating their system, what they're using, that they have somebody on the phone with them. And the second thing is that even if you have somebody who is using methamphetamine and you really are pretty sure that that's it, I would advise also providing a naloxone, a Narcan kit to go along with whatever you're doing with people, to prescribe the Narcan and to make it available, and just to remind them that when they're using methamphetamine with or without an opioid, that it's good to have Narcan in the environment, and explain how to use Narcan. It's not like it's going to be helpful to them if they're overdosing themselves, but that if they have people around them using, co-using, to have Narcan available in case there is an overdose that's unexpected, you have the drug in the environment. So just a kind of a reminder, so that as you're working with your folks who likely are involved with methamphetamine, just to provide them with that Narcan kit. All right, we're going to just tour briefly through addiction concepts, and really want to focus on the fact that most people either don't use drugs or a stimulant or whatever, or they are using at a level that doesn't cause problems. And so for them, it's fun. As people continue to use, the fun goes away if they're using, and you have fewer and fewer people using because they get problems. And problems for people who can quit, they do quit, which, as you'll see in the next slide, not being able to cut down or quit is a marker of addiction. And then, continued use of problems, people will continue to stop using. But those who have a strong allegiance to the drug will continue to just use the drug, but mostly experience problems. And at that level, you've got folks who are mild to moderate substance use disorder or addiction. And as you hit into severe addiction, you're really talking about a small number of folks who use these substances. So why am I saying this? It's because this process happens in real time. People don't really experience the shift from having more problems to most problems, because if they can't quit, they don't notice the subtle ways in which the disease of addiction continues. So, it's good to see this. It's analog. This is how we define people with addiction, though. It's the DSM-5 TR. And you can see here, these are how you measure. It's all behavioral. There is no biomarker here. And we measure severity as mild, 2 or 3 criteria, moderate, 4 or 5 criteria, and 6 or more being severe. I'll leave this on your own to review, but it's important to remember that's a digital approach. And there's a disconnect when we bring our digital thinking with our patients and analog, and we often don't bring them along about how it is that we come up with the idea of a diagnosis here. People really do appreciate if you break this down with them. So in methamphetamine, people have different kinds of patterns of use. These are typical. We've written about this. These are the weekend warriors where they begin using methamphetamine. It has a long half-life, 9 to 12 hours. So they start on a Thursday night. They dose again on Friday night, Saturday night. They take little blue pills along with methamphetamine to make sure that the sex that they're engaging, they can perform adequately. On Sunday, they may smoke out and with some weed, and then maybe even help themselves find their way to Monday using or Tuesday with a benzo. This is what it looks like for the shift workers. I like this approach because it shows you that if you're using a 1/4 gram a day or a 1/2 gram a day and you're dosing twice a day, this is what your system is exposed to. It's pretty frightening. If you were a neuron, you would be upset. And if you were in the cardiovascular system, you might have all kinds of pro-inflammatory responses. Here's the once in the blue moon kind of guy, every 6 months or so, every 3 months, methamphetamine gets used. And it's fairly, you can see that the dose levels of all of that are really different across the different ways in which people use, and that's important. From a system level as also from a treatment level. In terms of what we know about methamphetamine and HIV, that methamphetamine use increases incidence in MSM. It's the attributable fraction over the years still is high, but earlier on it was between 16 and 33%. So Explore is the left bar, Max is the right bar. And we also have seen that in data from HPTN showing that if men who have sex with men have social networks, that those social networks actually reduce significantly the incidence of HIV. And you can see this in here. Let's see, which one is it? It's right in here. The numbers of folks who have the number of social network size is actually predictive in terms of percent. So it's a very cool idea here that you can begin to see social networks as reducing risk. And this is the slide that shows the cumulative exposure. What we also have shown over the years, this is data we published in a couple of years back, showing that just the frequency, if you measure none, monthly or less, or weekly or more, so 0, a little, and a lot. So the zeros are circles, a little is squares, and the diamonds are a lot. You can see that the odds of having any of these awful conditions like unemployment, housing instability, blah, 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 you can read through here, that there is a stepwise reduction in terms of the odds from level of exposure with methamphetamine from none to some to a lot. And you can see this maps along. You can see also in here that this also happens for HIV outcomes. As we know, folks who are using a lot of methamphetamine tend to remain viremic no matter what we do. And that's the source of things like ACTG 5359 to be able to help folks to reach and sustain viral suppression. But we also see that people in here are less likely to take, we don't have the PrEP numbers in here, but it's the same sort of thing. People on PrEP using methamphetamine are less likely to stay involved in their regimens. What we also know in here is that there is no sort of dose response here in terms of HIV and methamphetamine. So if you're a man who has sex with men and you're using methamphetamine at all, the number that is serious is a stable 60 to 65% likelihood of being HIV prevalent. And we still see within Los Angeles in our cohort study an incidence rate of minority MSM of somewhere between 3 and 5% per year. So it's still a very important driver of HIV transmission. So after 30 years, we have some data that shows that we have medications with evidence to treat stimulant use disorder in primary care, in treatment settings for addiction, and also for clinicians who are working in infectious diseases. this practice guideline starts by recognizing the value of contingency management. We're in California. We can get contingency management into almost any clinical setting. It requires a lot of work within the infectious disease setting to be able to manage the needs of how you have people returning to clinic frequently to provide urine samples. But if contingency management were a medication, it would be a standard of care. It has a significant and high effect size. You can see the D's through here. The D's indicate the effect size of contingency management across meta-analyses. It's all regularly between 40 and 0.4 and 0.6. So I cut my teeth doing this sort of work with contingency management. I apologize to those who may have seen this presentation before, showing that even just providing the contingency management with no cognitive behavioral therapy, any talking to person or whatever, we could see that that itself was significantly better than a cognitive behavioral therapy in producing negative urine samples 3 times a week. So it's from 0 to 48. That's the possible, using just the simple contingency management. When you combined it with the cognitive behavioral therapy, you got a boost up here in terms of being able to produce even more negative urine samples. So this treatment, the therapy of contingency management plus a combined talk therapy was most effective. And you can see here that even just providing the contingency management alone at 12 months later across all randomized participants, it was equally likely to the talk condition or to the CBT or to the combined um, uh, CBT and contingency management in terms of being able to produce a negative sample at a year later. We got the first indication that if you reduce methamphetamine use among gay and bisexual men with methamphetamine use, methamphetamine dependence, you got a twofer. They reduce their unprotected receptive anal intercourse. So that's a really good first set of data showing that addiction treatment can function as HIV prevention. It also allowed us the opportunity to develop culturally tailored, specific cognitive behavioral therapy. These materials are online, and you can download them if you like. And it's a standard cognitive behavioral therapy model that is all gayed out. So all of the references within the manual are housed within the context of gay culture, which really is an important way of showing cultural competence when you're working with your participants and your patients. In terms of medications that we have, in terms of addressing meth use disorder, Maduka Trivedi and I published this study in the New England Journal. This is the extended release naltrexone, which is 380 milligrams. It's once every 3 weeks. And then the bupropion, 450 milligrams each day. We call this the combination. Produced a 11.1 percentage point difference between the medication and placebo, which is a number needed to treat of 9 at 12 weeks. I think it's kind of fun to look at the design here. I'm not going to spend time on it, but I do want to point out this is a novel design. It's a sequential parallel comparison condition design. So you have 1 to 3 assignments. So 1 active, 3 placebo at baseline. And you can see the nice medication effect. Here's the outcome period, the last 4 urine samples collected during weeks 5 and 6, because it's twice weekly. You can see that very nice medication over placebo effect. And then at the end of week 6, those who were not performing well in the trial were re-randomized 1-to-1 active to placebo. And this group here, you can see, moves forward, and you can see that the placebo continues to be at the floor, and there's a slight medication effect that you can detect here. So overall, it's 11.1 percentage points, but it really shows you that there is a robust effect of the combination over placebo. Something to think about when you're working in clinic. These medications are available on the formulary for Medi-Cal, so it's something to think about if you're not already doing it. Jeremy Kidd, a postdoc or a fellow from Columbia, NI published these data showing that if you take the combination, the adapt combination, and just look and see what happened to men who have sex with men compared to heterosexual men, we can see that the treatment effect was even more enhanced than it was for the heterosexual men. And this is, like I say, this is 0.15 almost compared to the overall difference in the trial with all participants of 11. We don't know what that's about. Jeremy put an R01, got a great score on it, and he's currently at NIDA Council, so we're fingers are crossed that he'll be able to do this study as a replication trial. We don't know yet. In terms of an alternative approach, Grant Colfax and Philip Coffin and their team have done a lot of work in terms of looking at mirtazapine 30 milligrams a day. Here's the data showing this is testing positive for methamphetamines at each of the weeks, and this is the drug line, and you can see it goes down over 12 weeks. In the initial trial, this was 60 people. It was a small trial that Grant Colfax did, but at the time, this was the only trial showing a significant effect in methamphetamine treatment. So this was hailed as a really great thing, but it was a small sample. Grant Colfax actually got funded to replicate this finding and saw that the effect size is about the same. This slope is about the same as this one here over the 12 weeks of the medication of 30 milligrams of mirtazapine daily. And you can see that's moving forward. And I will point out that Rebecca McKeaton has just finished a 400-patient trial of mirtazapine 30 milligrams a day in Australia, and findings will be broken. I don't think it's going to be CPDD this year, but the findings will be coming out soon, whether using a far less stringent approach to prescribing mirtazapine would produce significant improvements in terms of recovery from methamphetamine, because they're not using urine as an outcome, they're using self-report. So I'm not going to spend a lot of time summarizing, but the point here I want to point out is that the medication, the point that I really want to focus on practice change is to consider medication as a foundation of treatment for stimulant use disorder in infectious care settings. So this is the foundation. What we build on the foundation can be all kinds of things, and that can be part of that integrated strategies that I opened the talk with today, to think about things that we might do, including housing interventions, mental health, social determinants, addressing social determinants, treating comorbidities. And speaking of comorbidities, since I brought up the question, what's happening really in terms of the body? Because I really started with let's do whole person thing. Well, methamphetamine affects the cardiovascular system as well. And what we see in terms of, in heart failure groups, methamphetamine heart failure, hypertension is higher in that group compared to non-meth heart failures in meta-analysis in some studies, and meth controls in others. We can see lower rates of cardiovascular comorbidities in this group, largely because they're younger, and that among patients with heart failure, those with meth heart failure are more likely to have problems with inconsistent housing. So we also know that there's a lot of the methamphetamine is a sledgehammer in the brain. So it works on presynaptic neurons by changing the way in which dopamine is taken back up into the vesicle. It blocks the reuptake of dopamine, and it also will displace dopamine from the vesicle, causing neuronal damage. So that happens. So what we really want to focus on as we think more in this next decade of work is not just about HIV prevention, but also what's happening in the cardiovasculature, what's happening in the brain. So I'm not going to spend a lot of time with this, but I'm going to walk us through a few slides here. One of the things I've learned most recently is that methamphetamine use accounts for about 50% of non-idiopathic pulmonary arterial hypertension cases. So it's a fun little story on the right. I've left my slide deck for you, and you can look this up and read it yourself, or you can just read the little synopsis. But what has been known for a long time is that consistent exposure to stimulants is a primary cause of pulmonary arterial hypertension. It started with a Minorex, which was a stimulant that was prescribed for women for weight loss in the 60s in Europe. And in US in the late 80s and early 90s, I recall the whole fen-phen crazy. Women, and largely women, were using a lot of this fen-phen, and we started hearing reports of people, of these women who use fen-phen showing up with incident cases of pulmonary arterial hypertension. Even 3 months of use of phentermine and fenfluramine, would cause a huge increase in risk for pulmonary arterial hypertension. So you can see that odds ratio and the confidence interval is really wide, which lets us know that this isn't a common thing that happens, but in those who have this disorder, it's very well documented. I think this is really helpful also to look at. If we look at methamphetamine associated pulmonary arterial hypertension, it's much high. This is idiopathic pulmonary arterial hypertension. Methamphetamine rules the day in terms of pulmonology clinics here in the West. So one of the things to do to take home from today is to talk to the pulmonology folks about what's going on with your patients when they show up there with methamphetamine associated pulmonary arterial hypertension. So you can see over here in the East, this is the lowest rate, and still lower in the Midwest. And again, if you look at treatment assignments here in the West, we see that most of the drug-related treatment admissions over the years has been for methamphetamine, as opposed to in the rest of the country. So, one of the things that happens in pulmonary, why do we need to bring our colleagues into these discussions? Because people who treat pulmonary arterial hypertension have some of the same sort of stigma that we've been addressing here in infectious disease settings and in addiction medicine settings and in primary care settings. And that is the belief that people with methamphetamine use disorder or methamphetamine misuse are not going to be consistent with their medications. And this is a strong bias across medication and a strong bias across addiction medicine, that folks are not taking their medications. One of the things I've done over my career is listen to my patients, and reliably, patients with HIV would tell me, "Hey, Steve, I'm taking my medications. I don't know why I'm viremic or I'm having problems maintaining viral suppression." That led to a whole line of work with Jen Fulcher and some other folks here at UCLA, Cherie Blair, other folks working on, what's happening in gut regarding methamphetamine and HIV. And it's led to a whole line of publications about the direct effects of methamphetamine on the gut and HIV transmission. Fantastic stuff. But in pulmonary arterial hypertension, in the setting, the pulmonologist typically will require a lot of abstinence from methamphetamine before they'll begin talking about moving folks onto the parenteral treatments. And that's based on no data. This is where we can advise our friends and our colleagues that that's a bias and that we can do better. So these findings, like I said, I'm not going to spend a lot of time with this, led to a meeting in December 20th of last year with NHLBI and NIDA to talk about the ways that methamphetamine, HIV, and pulmonary arterial hypertension intersect and what we need to do to begin to bring light into understanding how we should respond optimally from a clinical perspective. This outlines what we're thinking about, captures much of the discussion that we had, but this is an area where I'm really focusing on working right now. In terms of where we are in terms of medication development for stimulant use disorder, there are no FDA-approved medications for stimulant use disorder currently. A small suite have strong evidence of efficacy, as I just reviewed, but most medications trialed have weak signals for efficacy. Even the combination, the emergency department folks call the combination of extended release naltrexone and high-dose bupropion interesting, but not really helpful. And they call the mirtazapine approach the metazapine approach. So it's, recognizing that we don't have something that's going to send everybody home happy. CS-1103 is the first medication that might actually provide some benefit. CS-1103, Clear Scientific 1103, is a chelating approach to methamphetamine treatment. It is an antidote to methamphetamine toxicity. The idea here is four medicinal chemists in Boston, who are my friends, had a family member who died from methamphetamine overdose, and they said, "Where is the Narcan for methamphetamine?" And the response back was, "Duh, nowhere." And so they said, "Well, we're medicinal chemists. We can design a molecule that can actually target methamphetamine in blood. Just hang out in blood. This molecule, all it does is a small molecule. It just hangs out in blood, and it's got a very strong binding potential, electrovalent charge on the molecule that when it meets methamphetamine in the bloodstream, actually attracts it, makes a complex, takes that complex to the kidneys, and it's flushed out of the body with methamphetamine and the CS-1103 not being metabolized. It does not metabolize in blood. It just gets moved out of the body through the urine. So CS-1103 does not cross blood-brain barrier. It just sits in the blood and it actually waits to capture methamphetamine and then take it out. So it's a chelating agent. And you can understand how that this medication is being developed for use in emergency department suites first. So it will be something like the Narcan for people who are coming in overamping or on methamphetamine overdose. I just want to focus on the fact that, what we have here is that methamphetamine has this very high rise over run in terms of plasma concentration, whether you smoke the drug or whether you inject the drug. And you can see here that the fastest way to get most methamphetamine into blood is with smoking, not injecting. So I'm going to stay focused on CS-1103, but this is an important finding that shows that people who use methamphetamine also have lower responses to medical care coordination. And these are data from LA County. Tens of thousands of patients in here showing that viral suppression is probability of viral suppression up to a year before MCC is going down. You start MCC, viral suppression odds go up immediately. They jump right away. And then, people maintain benefit if you have no comorbidities or even if you're just depressed. But if you are using stimulants or have housing instability, you're less likely to maintain suppression if you're compared to your peers. So that's an issue that we need to address. And CS-1103 may help here in the sense that these are non-human primate data showing the methamphetamine in urine 1 hour after a dose of CS-1103. So what they did is they took these non-human primates, put them in the lab, gave them lethal doses of methamphetamine, and you can see here, or near lethal doses, no animals were died in this. You can look and see that the red is the amount is saline and the blue is CS-1103 bolus. And you can see the amount of meth that's removed in urine after 1 hour is just outrageously higher in the CS-1103 than in the group that got in the animals that got saline. You can see here that it's a twofer. This is a rat study that was done. Now, one of the things about fentanyl and methamphetamine is those molecules are somewhat similarly shaped. And in fact, the binding potential for CS-1103 with fentanyl is a little bit stronger than it is for methamphetamine. So it works similarly for methamphetamine as it does for fentanyl. How it works is it hangs out into bloodstream, fentanyl comes along, and it actually makes the complex, and the drug is excreted unmetabolized. So you can see here what happens. These are ventilation. This is percent from pre-fentanyl baseline, and you can see that ventilation, once you administer fentanyl in the rats, is going down, down, down. And then here comes the study drug in this period, and you can see a nice dose response here in terms of CS-1103 in the rats in terms of the return to normal breathing. And you can see it's like very quick. This is 20 minutes, 22, 24, 26. And you can see that within a few minutes, like 4 to 5 minutes, you have returned to respiration about the level it was before the fentanyl administration in 30 rats. Fantastic data. And so the company is actually pursuing two indications with FDA, one for methamphetamine removal and one for fentanyl. And we're involved in that currently. We're doing this in a CRO. I'm helping guide the trial. But this is a blockbuster, folks. This is going to change our world, and it's going to change the way we treat our patients. I'm not quite sure how, but I really just want to focus as we start to end this talk about like some other evidence-based interventions that are currently available to manage methamphetamine use and treatment. Philip Coffin published this letter in New England Journal of Medicine evidence. And one of the things that Philip is looking at is the use of statins to protect the cardiovasculature in people who are actively using methamphetamine. Love the idea. It's a fantastic idea. And the idea of protecting cardiovasculature is something CS-1103 will no doubt actually help. But also CS-1103 will, by removing methamphetamine off the person who comes in with psychiatric conditions, who doesn't, will help people to resolve much quicker in the emergency department, and that's going to be a great benefit. So where are we with CS-1103? We are, the company has successfully completed the phase 1A study, first in humans at all low to high doses, and seen no treatment emergent adverse events. We are set to start our phase 2A trial the second quarter. So we're hoping like April to be able to start. We're not quite sure. There's some regulatory that needs to be resolved, but we're doing the work in a CRO as opposed to at the university because the CRO can get work done faster. So the questions that we have are, that we're really interested in. We're going to give people 30 milligrams of methamphetamine and then follow it up with, different doses of CS-1103 or placebo, checking safety. And we want to see, are there significant subjective and behavioral effects of immediate reversal of methamphetamine? We don't know. Would it cause precipitated withdrawal for people with methamphetamine? Well, that's not a concept, but would it cause precipitated withdrawal for those who use fentanyl? A very interesting question, because if you pull methamphetamine off board, if you rip methamphetamine out of the body, it'll pull the methamphetamine out of tissue, including neural tissue. What's going to happen behaviorally? We don't know. And what is happening at the opioid mu receptor when you pull fentanyl off board using this approach, because the fentanyl will be pulled off the receptor and taken out of the body using this complex from CS-1103. It's a different mechanism of action than naloxone. So it's going to be interesting to see what happens in terms of the potential for what's happening at the mu receptor. And we have folks who are looking at those sorts of things. Here's where we are. We're just getting ready for the phase 2 early efficacy trial. So you can see we have a ways to go before we'll be in the field. Jocelyn Kim and Jerry Zach are using CS-1103 with bar-coded HIV to examine the dynamics of methamphetamine in initial infection, and also in terms of treating the reservoir, the idea of being able to kick and kill this particular probe may be very helpful. Drug is currently on, it's not being shipped. It's actually in Jocelyn's lab now, and this study's ongoing. We're also meeting with Kathy Cahill and Chris Evans to understand what is happening at the mu receptor, with the idea that if the mu receptor is not altered dramatically, we might be able to use CS-1103 to co-deliver buprenorphine so that we could actually breach the discomfort gap that people have when inducting onto buprenorphine from fentanyl, which is a current problem. We also see, so the company has a laser focus on getting FDA approval. And then there's all kinds of really cool ideas that will happen after this drug gets ready. And the idea here, when it's marketed, is that we're going to jump on this multi-purpose technology to use buprenorphine with CS-1103 to induct patients, to integrate doses of ART for post-party recovery, and using it primarily as a way of advancing HIV PEP. And then the question about being able to have patient-controlled use of this drug could actually help people who are committed to changing their methamphetamine use, but occasionally have periods of return to use to be able to limit the outcomes of this, the negative consequences of a return to use. So where are we in terms of this work ahead? I don't know. The future is so hazy. Things are nuts here. Um, you know, it's a new day. Releasing levers on HIV and other infectious diseases will lead to poor health outcomes for HIV and will increase forward transmission. We learned that in COVID. We're going to see this again. We're just now in LA County starting to see incident cases reduce. And now this is happening. So we're just not quite sure what's going to happen in terms of release, if we're going to be able to continue working in the way we did. Probably not. Managing methamphetamine in HIV settings improves the health of the whole person. We want novel developments in treating methamphetamine moving forward because they present opportunities for multi-purpose technologies that meet the whole person and the needs that they bring to the setting of HIV and addictions. And then CS-1103, this is the most exciting work of my entire career, folks. This is a really amazing new drug, and I look forward to what happens next. So I'll stop here.
Great. Great talk. A few questions that came up along the way. Earlier on, you talked about some of the social networks and how it would have a positive effect. I don't know, Jeanette, if you're still on, if you wanted to unmute and ask your question.
Yeah, hi. Thanks. That was a great talk. I was just thinking about the many patients I've had over the years who use methamphetamine and the conditions that create such use. And so, a supportive social network is often protective, right? So when people move back home or move back with people who are not using, but I was interested in your social network discussion because many of my patients, I have a couple of patients who have become unhoused and then they start using meth because that's their social network. So I wanted to hear a little more about that.
Great question, Jeanette. I mean, as you know, one of the hardest things that people who use methamphetamine regularly will trim their non-drug using social links. They just do. And part of it is it's a two-way phenomenon where the people in the non-drug using social network say, you're too much for me. I can't deal with you. I need to push you out. So when a person actually stops using methamphetamine, as you know, all they have are the drug using social connections. So one of the things we tend to do is to try to help people to make those pro-social non-drug related connections, which is really hard work when you're in early recovery from methamphetamine use. Actually, it's really hard work. You can put an end of the period at the end of that session, even sentence. Even for me, it's kind of hard to develop new connections. So that's just the issue. For those who can do that, though, it is protective, and it's protective at a significant level. So the idea here is just to continue to promote people finding ways to get out of their drug using environments and behaviors and to trial new activities. So one of the things I will do, because especially with gay men, because gay men define themselves by having sex with other men, while they're in treatment with me, I will say, what I want you to do is before I usually see people every week. Before I see you next week, I want you to go get laid without using methamphetamine. So the idea here is it's a controversial idea, but it really helps because if you're defining yourself as a gay man because you have sex with other men, and your problem is that methamphetamine sex causes you to go crazy, then if you take methamphetamine off board, what you have functionally is an eighth grader in front of you. So the idea here is you want to be able to help people to re-enter the world of being a sexual being and being able to be a social being and to be there to support them early on and consistently.
Great. A couple of questions about GLP-1 agonists, what's the existing evidence for those?
Great question. The existing evidence for GLP-1s for for stimulants is non-existent. It's not non-existent, it's just weak. So there's some data being used with GLP-1s for smoking cessation, and that seems to have some effect. The cocaine data is basically useless. Right now, the best data for GLP-1s are for alcohol use disorder and opioid use disorder. So it's a bit further to go to stimulant treatment. Now, we've been talking about a design for that, just to do a small study and do a Bayesian analysis, maybe even an uncontrolled study where you predict the number of people you need to treat to show a success in a phase 1 trial with phase 1-ish trial, but it's still early days.
Anik, you want to ask your question?
Yes, it was just a follow-up of the social network question. I was wondering if there was anything like that happening in apps for finding sexual partners, kind of saying in the same way that you can say you're HIV positive or negative or whatever, also say I'm off meth, to kind of link with other people who are also off meth or
It's
who
not
want to.
there, and I'll tell you why it's not there, because people lie. So, like the idea of, this has been my experience with my patients is, they go to find this. That's why I prescribe this early, is that, you want to have people fail, and they will fail on this task of getting laid without using meth, because they show up. And I've heard this response multiple times. I showed up, this guy says, I don't party, I don't do Tina, I don't, whatever it is. And I show up and the little baggie is there. So that's what happens in real life. So the issue of, being able to build a support structurally is not viable. This has to be something that you instill in people to be able to manage, because methamphetamine in the presence of somebody in early recovery or early abstinence is there's nothing we have that has an effect size that can intervene with that gap, that person at that moment. So it's got to be inside here, and it's got to be routinized and practiced.
Thank you.
And then questions about what must have been fascinating discussions with regulators and IRBs for starting the studies. So if we understood correctly, you're administering methamphetamine to people who are not methamphetamine users.
No, these have to be methamphetamine
Okay, great.
dependent folks.
That makes a bit more sense. So,
But even still, we considered giving people Digoxin, but we just thought that the IRB would not go for it. But we didn't bother to talk to the IRB. We just thought reviewers wouldn't like it, regulators wouldn't like it, and so I was interested in the hurdles, the walls you faced, if any, and sort of getting to that point.
You said it best, Scott. Start the conversation with your IRB. So this is what we've done over the years at UCLA. We've done a number of these phase 1b trials where we bring people in with methamphetamine use disorder with a test agent, and we dose them with the methamphetamine. Then we bring the test agent on board or placebo, and then see what happens. So the idea here is, you want to know, the FDA, if you're going to go forward, you need controlled data of what the drug-drug interaction is. So what is the likelihood that your new test agent is going to invoke a cardiovascular primarily or other sort of effect? DEA. No, DEA doesn't care. We administer methamphetamine.
They care more about cannabis.
That's right. They care more about like Suboxone and making sure that make it hard to get, or methadone or whatever. So
yeah, I'm sure a lot of interesting work to be done about just what's drawing people to participate in the research. What are they hoping for? Because I imagine there could be some misconception about what this is actually going to do for them. But I'm just curious what their motivations are. If they're interested in
that's a very good
recovery.
question.
Yeah.
On this one, I mean, we've always used the people with methamphetamine use disorder have altruism. So they really do believe that if they can use their illness as a way to bring health to someone else, that's an important process to get involved with. And of course, we pay them well. So the idea of both of those factors are driving people to come in. It's hard to find methamphetamine addicted people with elite physical health. Most people coming in will have high blood pressure. They will have untreated high blood pressure. And so just getting people, we have really strict markers about how high the blood pressure can be before they can be in the trial. And we have really strong no-go positions where if somebody's um, blood pressure drifts up above like 150, um, over 100, then we have to stop the trial for that participant. So that's how you do this, Scott, is you actually do, and certainly you can use my protocols. It's fine. So I'm happy to share.
And then in a private message, someone asked about, do you have any advice for postdocs and trainees about how to weather the current storm, and in particular, any insights as to research lines or things that you think would be strategically uh, better choices?
Well, that's my Friday. That's what I'm doing today. So I think for the PhDs and postdocs, if they have clinical skills, they'll weather this easier than if they don't. The pure scientists are going to struggle. There will be people who are not here next year who we know, because they're going to need to find something else to do. We're also firing so many white collar people. It's going to be hard to find relevant positions in labs, because outside of pharma, it's really going to be tough. So what we are doing is talking cold reality with our people and saying, if you have a plan B, take it. This is not the time. This is like when Reagan did this to science, to research, when he started in 1980. It was a little earlier than I came in around 86 or 87. So we were coming out of that, and largely due to HIV/AIDS, where Reagan was forced to actually restart research. I don't know where this is going to go, but this is not a good time for people to be starting basic science careers, especially in the areas that touch gender, transgender, sexuality, um, sin.
Yeah. I mean, who can tell? I mean, from what we've seen, we haven't really I haven't seen a direct targeting of research about people who use substances. And certainly there's been a lot of bipartisan support, so we can, perhaps in vain, but we can hope that that line of research is
No,
relatively untouched.
exactly, Tim. I think this is going to be more of a structural issue.
Yeah.
The issue that so everybody on the call today, the one thing I will leave you with is to call your PO. If you have a PO, call them today. And the first line is, how you holding up? That's all you need to do. And then just talk to them. These are humans. These are people who are science focused. They're either scientists or they're interested in making science work. We have to preserve NIH as the first round of defense for those non-clinical PhDs and scholars who are doing basic science. And we do that by taking care of our POs. So do this. I've done this repeatedly over the past couple of weeks, and it's been really something that I felt good about doing because I was able to support them. And they will tell you what they need you to do from the outside to help them. So please do that.
Great. Steve, thanks so much. Great talk. An incredible line of work. And my first week in Hillcrest Hospital, I'm kind of floored by the extent of substance use affecting the patients in that hospital. So it's been really interesting to see. And there's other in the chat, people confirming their kind of the biases of pulmonologists for treating pulmonary artery hypertension, and a lot of the same experiences that you've talked about. So thanks again for joining, everyone. Hope you have a good Friday. Have a good weekend. Stay strong.
Thanks, Tim.
Thanks.
Really great way to start the day, guys. Awesome.
Thank you.